Welcome everyone. It is Dr. Lindner, and what we're going to be discussing in this video is going to be tissues. And you'll see that we have four different types of tissues here. We have epithelial tissue, connective tissue, muscle tissue, and nerve tissue. These are the four classifications of tissues. Now, if we were to just look at, in this picture, we have an arm, and on the top and on the bottom where I have numbered 1 and 2, it's only showing two of the four types of classifications of connective tissue. And in number 1, it says epithelial tissue, where it lines the surfaces in the body. And here it's showing muscle tissue. Like this looks like the biceps brachii. So actually, let's see if we can name this number 3. And then over here we have nervous tissue, which is number 4. So this picture is showing the four different types of tissues. Epithelial tissue, number 1. Number 2, connective tissue. Number 3, muscle tissue. This is one example, the biceps. And then neural tissue over here, number 4, like different nerves. And under connective tissue, it shows four different subcategories underneath connective tissue. It shows bone, it shows cartilage, it shows blood, and loose connective tissue. Now, I'm not really getting into much detail just yet, just showing the significance of the four different types of tissues and that it pretty much can be found everywhere within the human body. So again, how do we get tissues? Remember, tissues are when a group of cells come together that has similar structure and function, and when those cells come together, we get tissues. And if you remember from the readings within the first week, cells can come together by different types of junctions. Cell junctions like tight junctions, gap junctions, desmosomes, hemidesmosomes, adherens junctions. Those should sound a little bit familiar. So again, the four types are epithelial tissue, connective tissue, neural tissue, and muscle tissue. And these classifications of tissues, each one have distinct structure and function, right? Structure and pattern basically means the same thing, and then function. So structure determines function. And tissues are organized into organs, like the heart and the kidneys and the lungs. The arrangements of tissues in an organ are going to determine the organ's structure and function. When we study tissues, we call that this histology. Remember, when we studied cells, it was cytology. When we studied tissues, histology. We're going to look at the first type of tissue, which is epithelium. And you'll hear the word epithelial, epithelium. They're the same thing. Okay, now epithelium or epithelial or epithelia, these tissues are going to act as a lining type of tissue or a covering. Okay, well, what's it going to cover? Well, it can cover the external surface of the human body. And when I think of something that's covering the external surface of the body, what comes to mind to me is going to be the skin. Okay, and when I think of the skin, we think of dermis. Dermis means skin, and epi means above. So we have epidermis, which is the lining of the skin, but it is the superficial part, right? The epidermis is the part that you can see when you look at someone, but the dermis is going to be a little bit deeper to that. So we have the epidermis on top and the dermis below. When we say epithelium can line, these are going to line body cavities. We spoke about body cavities last week, and tubules. Now, within the word tubule, we see the word tube. So this is a tube, right? And we have lots of these within the human body. Maybe they're not straight, maybe they're curvy, right? Maybe we're talking about the intestines. That could be a tube. Maybe we're talking about little blood vessels. Those could be tubes. Tubes and tracts are the same thing. Tracts, like the digestive tract. That's a C. The digestive tract, the respiratory tract, right? Everywhere we have tracts, we have tubes. In the respiratory tract, we have the trachea, right? That's in the throat. It's just another type of tube. So anywhere we have tubes, we're going to have epithelium. Just keep that in mind. Also, we have specialized epithelium that are called glands, and glands are lined by epithelium, like the sweat glands, your oil glands, your mammary glands. Okay, these are just a few examples. Now, when we look at epithelium, one of the things that I want to mention is that epithelium is made up of a bunch of cells that are closely packed together, like this, right? I have 3 cells with a nucleus here, and I'm going to do double layer, why not? So these cells are very closely packed to one another. This is looking at them under a microscope, compared to maybe, let's say, red blood cells, which may be scattered a little bit further apart. Maybe this is a white blood cell with a nucleus that looks like that. These are red blood cells. This is a white blood cell. And I don't know, let's make another white blood cell here with different shaped nucleus. Okay, so these cells are closely packed together. These are spread further apart. Okay, when we see cells that are spread further apart with lots of fibers in between and protein fibers and extracellular matrix, this term extracellular matrix, this stuff is seen in connective tissue. Connective tissue is when you have cells that are further apart from one another, and epithelial tissue is when the cells are really closely packed to one another. And here you have very little extracellular matrix, but over here we have lots of extracellular matrix. Lots of stuff between the cells, right? Extracellular, extra, outside of the cells. Okay, so now that we mention that, when we look at epithelium under a microscope, or when we look at um, these cells under a microscope, we have to ask ourselves a few questions. One of the questions, if it's epithelium, right, the first question we would have to ask ourselves is, are the cells closely packed together? And if they are, we know it's epithelium. If it's not, then it's another type of tissue. Maybe it's connective tissue, but it can't be epithelium. So once we say, okay, the cells are closely packed together, it's epithelium, the second question we can ask ourselves is, how many layers of cells do we see? So here's one layer, and here's another layer. So we see 2 layers, one stacked on top of the other. So is it one layer or is it more than one layer? If it's one layer, we call it simple epithelium. And if it's more than one layer, it's stratified. In this case, we have more than one layer. So it's a stratified epithelium. The next thing we do, the third question we ask is, okay, what's the shape of the cell? Is it cube shaped like the ones up here that I drew? And if it's cube shaped, we call it cuboidal. Are they column shaped? Are they tall rectangles like this? Right? If they're tall rectangles and they're columns, then we call them columnar. If they're cubes, like squares, then they're cuboidal. And if they're flat and irregular shaped like this, then we call it a squamous cell. Okay, so those are 3 questions you would ask. So when we look here at this from the side view, we see that this is 1 cell layer thick, this is 1 cell layer thick, and these are 1 cell layer thick. But over here and to the right of that, these are multiple layers. This is more than one layer. This is two layers. And this also is two layers. So once we see that they're one cell layer thick, we call it simple. And all of these in this column are going to be simple, right? We see simple, simple, simple. In column number 2, up and down, we see stratified because they're more than one cell layer, right? So that was the second question we asked. What's the shape? They're closely packed together. That's the first question. So we know it's epithelium once we see cells closely packed together. And then the second question we would ask is going to be, what's the shape of the cell? If they're flat and irregular, it's squamous. If they're square or cube shaped, it's cuboidal. And if they're tall columns with a nucleus, then they're columnar. So here we have simple squamous epithelium. To the right of it, stratified squamous epithelium. Here we have simple cuboidal epithelium. To the right of it, stratified cuboidal. Why are they stratified? Because we have two layers of them. Then we have simple columnar, and then we have more than one layer, so it's stratified columnar. One of the things I want to point out is that many times with columnar, we have this layer on the top that are made up of a bunch of cilia. And cilia are these hair-like projections, and they can sweep in this direction, or they can sweep and move and sink in that direction. Okay, almost like a toothbrush, right? Like those bristles can move in either direction. So keep that in mind. We'll see that typically with columnar cells. Now off to the lower right, we see that there's a term here called pseudo stratified. Now the word pseudo means false. So we're saying that this is a false stratified layer because if we look to the left of it, right, all of these nuclei are nice and aligned, right? So when they're nice and aligned, we could see two layers or one layer. But here, the nuclei are scattered at a few different levels, so it gives the appearance that perhaps this is more than one cell layer because the nuclei are scattered at many different levels within the columnar, but it's still one cell layer thick, so we call it pseudostratified columnar epithelium. And again, when we see these, whether it's pseudostratified, when we see these columnar, we're typically going to have these cilia. And this is kind of important because let's see if I could draw this here for you. Let's say this is the windpipe. Let's say this is the trachea. Okay, this is a tube or a tract. And then let's draw these columnar cells here. And let's put one here, a nucleus. We'll put another nucleus here. And how about we'll draw, let's just try and do it quick. So we'll draw a bunch of these columnar cells on this side. These are columnar. And then we'll do a bunch of other columnar here. And we'll put the nuclei scattered at different, so really this will be like a pseudo-stratified layer, okay? How about that? We'll put a nucleus here, nucleus here. So because these nuclei are scattered at different levels, it gives the appearance that it's stratified, but it's really not. Okay, and then I'm gonna put, let's put the cilia here. So we have these hair-like projections. We have the cilia coming off on both sides of these pseudo-stratified. Okay, so here is cilia. And what did I say about cilia? I said that they're going to be beating rhythmically. And these are going to be beating upwards, right, in this direction, upwards. And I'm going to put another important type of cell. We'll make this one green. We're going to put a green cell here, a green cell here. These green cells are called goblet cells. So G for goblet, I'm using G for green. These are goblet cells, and what goblet cells do is they produce mucus, and the mucus is going to be sitting right here on the top of the cilia, right? So these goblet cells produce this mucus that move up to the surface of the cell. It's going to release it through exocytosis, and it's going to stick to the top of the cilia. But remember, the cilia are beating superiorly. Now, you are breathing in debris, right? You're breathing in, let's make it blue. So you're breathing in air this way, full of dust particles, maybe smoke, maybe pollution. Maybe you're a smoker, and maybe you're a smoker that's inhaling all these carcinogens. And what that does is it actually paralyzes the cilia. So they're no longer cilia here beating rhythmically upward. There's nothing there for the mucus to stick to. And then what ends up happening is these pseudo stratified ciliated columnar epithelial cells, they die off and they end up becoming squamous cells. They'll become squamous cells. So from pseudo-stratified, you get these squamous cells, and when one cell changes shape to another, there's a word for that called metaplasia. Metaplasia is typically precancerous. So you get this condition, metaplasia, when one cell type changes into another. And when you get squamous cells here, we call it squamous cell carcinoma, right? Carcinogen. Carcinoma is cancer. So that's throat cancer. That's from smoking. And when these cilia get paralyzed, what ends up happening is when the person wakes up in the morning, they do this. Right? They get this smoker's cough where they have to force all that debris upwards out of the lungs because they don't have the mucus there, they don't have the cilia beating rhythmically to help protect them. So there's some clinical relevance to tissues, and then what happens when they die off, metaplasia, typically precancerous. Women know this better than guys because when women go to the gynecologist, they'll slough off cells of the cervix, the tip of the cervix, right? Or the vaginal cells, they'll slough them off. And oftentimes it comes back and they'll tell the woman you have cervical dysplasia, which is the same thing as a metaplasia. And they'll monitor and watch, especially in instances where they think there may be HPV, the human papillomavirus. Okay, we know that there's 4 different cancerous types um, of HPV, and then some are high risk and some are low risk. So again, when you see cervical dysplasia, it could be seen with HPV, or it could also be seen in women who are taking the pill. And when women take the pill, it depletes them of B6, B9, and B12. Okay. And when you have the deficiency in B9 or folic acid, commonly you can see dysplasia as well. So in my practice, when I look at patient intake forms and a woman is on the oral contraceptive, I'll typically put her on a good B complex. I'll never put her on folic acid. That could be very cancerous to many women if you give them just B9 in the form of folic acid. So typically the methylated form is better. It's called methyl tetrahydrofolate. Okay, so let's take a look, just another picture showing the same type of cells. Again, it's one cell layer of these flattened cells, so we call it simple squamous epithelium. When we have simple squamous epithelium, it's a lot easier for things to diffuse, to pass through something that's one layer, and easy for things to pass through in this direction through one layer. So maybe we're diffusing oxygen and we're trying to get rid of CO2, carbon dioxide. So we'll find simple squamous in the lungs. Below it, we got a few layers of squamous. So it's stratified squamous epithelium. Now we need several layers. It's stratified, much more important for protection. So we'll see this in the skin, especially the dermis, the epidermis. Okay. Then off to the right, we have these cube-shaped, it's one cell layer, simple cuboidal epithelium. We'll find these cuboidal cells in the kidneys, in the part of the kidney called the nephron. When you get into anatomy and physiology too, you'll get into the kidney, you'll learn about the nephron. But in the nephron, we have a proximal convoluted tubule. There's the word tubule. Then we have a distal convoluted tubule. And both in the proximal convoluted tubule and the distal convoluted tubule, we're going to have simple cuboidal cells. Down below it, more than one cell layer of these cube shells, so it's stratified cuboidal. Off to the right, notice the nuclei are pretty much at one level, so it's simple columnar, and then down below it, the nuclei are at multiple levels, but it's still one cell layer thick, so we call it pseudostratified columnar. And these are the ones that we said will often have the cilia on the top. Okay, we'll find it in the respiratory tract. A final type that I haven't mentioned yet is called transitional. Transitional, think of a balloon, okay? If you have a deflated balloon, if it's deflated, let's think of it like this, and you have this deflated balloon like that, and you put the letter, I don't know, let's say you draw a box on it like this, a square with a marker, and then you blow that up, right? You keep filling it up with air. Eventually, the square, what happens? It may end up looking round when it's blown up and stretched out. So with transitional cells in organs and parts of the body where it can alter and change in their shape and size, like the urinary bladder, if it's filled with urine or not, if you have an empty bladder, the cells can alter in their shape. So it's not just cuboidal, it's not just columnar, right? You'll see different shapes. That's transitional. Let's look at some of the functions here. This has shown up on some of the McGraw-Hill Connect quizzes in the past. So when we see simple squamous, right? So now this is just one cell layer thick. This is important for diffusion. We said like for oxygen or for CO2 to pass through that one cell layer thick. If it's one cell layer, it's easier for filtration and secretion. So that's simple squamous. When we look at simple cuboidal, I think of the kidneys, and the kidneys are important for absorption, secretion, as well as protection. That's simple cuboidal. When we have stratified squamous, now we have multiple layers. So this is for protection of the underlying layers. Because you have more than one layer, it's stratified. We think of the epidermis, right? We think of the skin with that. With pseudo stratified, this is like the trachea picture. We have epithelial functions in the movement of secretions like mucus. What was it that produced the mucus? Remember those goblet cells. Those goblet cells produced the mucus that moved across the apical surface, right to the top where those cilia are. Apical means top. Transitional epithelium, this accommodates the extension and relaxation, right, with changes in organ volume. You think of the urinary bladder, anything that's urination, urinary bladder, ureters, urethra, all going to have transitional epithelium. I mentioned cilia already. These are long, slender extensions that come off the cell membrane, and they are designed to move secretions and fluids across the surface, just like mucus. We have something smaller than cilia, and these are called microvilli. These are much smaller projections, and they're designed to increase absorption. Where we have microvilli, let's see if I can draw this way. We have, this would be, let's say, the small intestine, part of your gastrointestinal tract. And off of the intestines, we're going to have lots of microvilli. Again, they're going to be on both sides. And these microvilli are designed to absorb nutrients, the absorption. Think of it as a sponge. It's going to pull in nutrients and move those nutrients into your blood vessels. The carbohydrates, the proteins, the fats, right? Everything that you're consuming through food is going to be broken down by these digestive enzymes in the gastrointestinal tract, but then absorbed into your bloodstream so that your blood can bring it to other parts of the body for fuel and nutrition. But you have people that may have celiac disease or sensitivity to gluten, in which these microvilli break down. And if these microvilli break down and they're not functional, then the individual becomes malnourished because they're not absorbing the proper nutrition, right? So that's why many people are gluten free, so that they can grow back these microvilli. Okay. People who consume foods that they are allergic to and they know they're allergic to, but they eat it anyway, they're typically going to get, like colitis and an inflammation of their stomach and intestines because they are breaking down these microvilli, creating inflammation of the belly. Okay, so again, let's do a quick little review here. So when we look at epithelium, we got to look at the number of layers. Are they one cell layer? We call it simple. Are they multiple? We call it stratified. Then we look at the shape. Is it squamous, cuboidal, or columnar? If they're squamous, we know that they're flat shaped, and we use these for protection, secretion, or diffusion. We think of the lungs when we talk about diffusion of gases like oxygen and carbon dioxide. When it's multiple layers of protection, we think of the skin. With the cuboidal cells, we think of the proximal convoluted tubules of the kidneys. And then also some glands, like some of the sweat glands are going to have stratified cuboidal. And then columnar, we're going to find those along the gastrointestinal tract and in the respiratory tract. We mentioned that pseudo-stratified, it looks like it's one cell layer, but it's not, because the nuclei are at different layers. I'm sorry, it looks like it's multiple layers, but it's really not. But it gives the appearance that it's multiple layers because the nuclei lie at different levels. It's really simple columnar, right? So pseudo means false, false appearance. And often where we have pseudo-stratified, we're going to see ciliated, especially in the respiratory tract. Transitional, remember we said these are many different shapes. We think of the urinary system, whether it be the urinary bladder, the ureters, and even the urethra. The cells can be flat when the organ is full, or they could be rounded when the organ is empty. So transitional epithelium, we typically see that in the urinary system. So let's look at last slide for epithelium. If we are talking about, let's look on the top of letter A with the lungs. In the lungs, where we have oxygen and CO2, notice it's simple, squamous. A lot easier for oxygen to diffuse through something that's one cell layer, and it's a lot easier for CO2 to find its way out. If we look at the bottom right corner with stratified squamous, now we have multiple layers of those flat cells. We're going to find this either in the skin, we could find it in the esophagus, right? It's stratified squamous. We need protection over there. What happens if you swallow a cracker that has a sharp corner? You don't want one layer. It's got multiple layers to protect it. Pseudo stratified, ciliated epithelium, right? Pseudo stratified, ciliated columnar epithelium. These are in the respiratory tract. Remember, see all those cilia here on the top? We don't want those burning off. And then remember, we had those goblet cells that produce the mucus, and that mucus is like that sticky glue that's on top, and it can trap all the dust and debris. Then we have simple cuboidal. We're going to find that in the nephron at the proximal convoluted tubule or the distal convoluted tubule. And you can see here, here's the actual tube, and then these are just one cell layer thick of cuboidal cells. And each of them have a nucleus, of course. And then on the bottom left, we have simple columnar epithelium. These are found in the GI tract, especially of the intestine, and typically you'll have microvilli on the top of these to absorb nutrients. Okay, so simple epithelium, we have simple squamous, simple cuboidal, simple columnar, and under the stratified, we have the same three: stratified squamous, stratified cuboidal, stratified columnar, and then the two unique ones: pseudo, stratified columnar, and transitional. Good place to take our first break.